Thanks, everyone, and thanks to the organizers for this opportunity. It's good to see everybody in person. I have seen people and met people that I have only met on Zoom at this conference, and I will say the advantage to being on Zoom is that I could pretend to be taller than I actually am. But it is good to see you, both old friends and new. Today I'll be talking about the work that we've been doing on using MPS to model in vivo PBPK, and using this work to predict renal clearance in both healthy patients and people with disease, and also maybe with drug interactions. So we all understand why MPS can be useful, but why do we want to use MPS to predict pharmacokinetics? And there are three major reasons for that. One is that there is a high potential for suboptimal drug exposure, toxicity, and drug interaction, particularly in patients with chronic kidney disease, and that's the population that I work with. On the average, a patient with end-stage kidney disease may take 10 to 12 medications, 19 pills per day, and those are prescribed by 4 to 5 different providers. I recently talked to a patient who was taking 54 pills a day. So the potential for drug interaction in this population is tremendous. And so we would like to be able to model PK in patients with kidney disease. Now, this is a requirement by the FDA for drugs that are chronically administered, both small molecules and some biologics, and for drugs that are expected to be administered to patients with renal impairment or who are on dialysis. And this data, PK data in this population, is particularly difficult to obtain. There is basically an endless supply of graduate students who will take any number of substances for money, so we can get that PK data in healthy populations without much trouble. But getting that data in patients with kidney impairment is challenging. Not only are these patients medically fragile, they also often have a lot of comorbid conditions that make it very difficult to do a dedicated PK trial. And because of that, these kidney impairment studies are also incredibly expensive. You need to do a single-dose trial in 5 cohorts, both from healthy with different degrees of kidney disease all the way to end-stage kidney disease, and the estimate of costs for one of these studies is between $1.2 to $6 million just to satisfy the renal impairment requirement of the FDA. So if we can use MPS to streamline some of these steps, maybe we can get drugs that are safer and faster to the market. So to study these phenomenon, we've developed a roadmap using kidney MPS and PBPK modeling techniques to model human renal clearance. And this is the workflow that we followed. First, we use the MPS platform, then we scale that MPS data to human renal clearance, and then we can use that data to model PBPK. So first, I'd like to introduce our platform. We use the Nordis device. It's a homegrown company that came out of the University of Washington. It started as a fairly crude sandwich of polycarbonate with cast PDMS in the middle. There was a single flow path, the potential for transluminal flow, and some bubble traps. The next iteration included some thumb screw valves and self-sealing injection ports, but was basically similar to the single-channel device. What we're using for these experiments is a dual-channel device, which places 2 tubules side by side, one of which we use as a vascular channel to model the blood side, and the other we put in a proximal tubule, which is our kidney side. So here's what this looks like in real life. To remind you of our setup, remember we've got 2 side-by-side tubules. One we culture with the proximal tubule, and that I'm showing you on the right, and on the left we culture HUVECs. We culture those HUVECs for 1 day. We realize that's an immature HUVEC culture, and that's intentional. The renal vasculature is very different than a lot of the vasculature in the rest of the body in that it's extremely porous and extremely leaky. So we use an immature HUVEC culture that we grow for 1 day, which is still extremely leaky, but acts as a structural stent to maintain the patency of the vascular channel. The HUVECs, on contrast, we grow for 5 days to make sure that they're a mature, confluent culture and don't have any leaks or holes in that tubule. And this whole system can model the many steps of renal elimination. So we have the blood side, we have solutes or toxins or compounds or endogenous compounds that can extravasate out of the vascular channel. It can cross through that interstitial space and then be taken up by transporters in the proximal tubule or paracellular transport, or however that compound happens to transit into the urine. And then we can collect the effluent out of both the vascular channel and the tubular channel. So now that we had a system that we thought looked pretty good, We tested the system, we tested the system, I feel like I've spoiled the surprise. We've tested the system with both fitsidextran, which is a large molecular weight compound, radiolabeled mannitol, which is a small molecular weight compound but has high polarity and is negatively charged. It's often used as a marker for paracellular permeability across epithelial barriers. And we also used para-aminohippurate, which is a known substrate for renal transporters. We also perfused the para-aminohippurate with probenecid in the vascular channel and the tubular lumen to test whether or not that translocation was due to active transport and that transport could be inhibited by probenecid. And what we found was on the left is you're seeing what came out of the vascular channel, and we did that just to make sure that what we put into the vascular channel also came out of the vascular channel. And this is what Nasi alluded to. We wanted to make sure that our compound wasn't getting absorbed into the tubing or the PDMS or anything along the way. So what we put in was what we got out, which was helpful. And we also wanted to see what came out of the tubular channel. And what we found was that some of the fitsidextran came through, almost all of the mannitol came through, and that some of the PAH was found in the tubular lumen, and that much of that could be inhibited by the incorporation of probenecid. So now we have a system that has two channels, has a blood side, and it has a blood side, and it has a tubule side. And from that data, we were able to calculate a clearance from the blood into the urine. And so we had to figure out how to scale from a credit card-sized MPS to 2 human kidneys that are about the size, each the size of your fist. So fortunately, we were able to scale up the size of that tiny, tiny tubule on the chip into the length of a proximal tubule, the diameter of a proximal tubule, the estimated surface area, and multiplied that by 1,000,000, because that's approximately how many nephrons you have in a healthy human. Fortunately, we were able to obtain PAH clearance data from clinical study and compared our derived renal clearance data to that was derived from actual people. And remember that actual people are the original multi-organ in a body that's appropriately scaled. So we were able to translate that data from a chip into an actual whole human, and we found that the ratio of what we predicted to what we actually observed was about 1.96. So we were off by about 2-fold. And in the PK world, that's actually very, very close. In fact, when we submitted this for review, we were told that the value was so close that they didn't believe it. So we decided we needed to test, obviously, try it again with another substrate. So we decided to, with support of NCATs, to evaluate the secretory transport of morphine and morphine 6-glucuronide. We infused each of these compounds separately into that vascular channel of that dual channel chip in the presence and absence of an inhibitor cocktail of probenecid and TEA. And at this point, we had also figured out how to test the tier of the tubules, and we found that our tubule was actually quite, had decent resistance. We were about 150 ohms per meter squared. So we infused morphine and morphine 6-glucuronide into our MPS. You can see morphine on the left and morphine 6-glucuronide on the right. And in the dotted line is the vascular channel, again, to make sure that what we put in actually is what came out, so that our cells were exposed to the concentrations we thought they were. And on the bottom in that dark solid line is what came out of the tubule. Morphine on the far left with that inhibitor, and then with inhibitor on the right. So you can see there was a significant decrease in the translocation of morphine with the addition of the inhibitors. And also similarly with morphine 6-glucuronide, we saw a significant drop in the translocation with the inhibitor compared to without the inhibitor. So we took this information, we calculated the apparent clearance, we calculated the apparent clearance from passive diffusion, and then we calculated the apparent clearance by active secretion, and some of this was bolstered with some 2D transwell data. So here's what we have found for both PAH and our morphine and morphine 6-glucuronide experiments. I'm showing you the predicted clearance, renal clearance that we determined using MPS, and I'm showing you the clinically observed renal clearance that we see in patients. And what you see is that as we went into morphine and morphine 6-glucuronide, our predictions actually got a little bit better, and that all of our predictions were within 2-fold of what we actually see in human beings. So not content to declare victory, we took this project one step further and decided to use this data in a dynamic PBPK model. So with the generosity of Drs. Huang and Nino Isaharanen, who work just upstairs from us, we were able to use their full-body PBPK model to model morphine and morphine 6-glucuronide. For this model, we assumed IV administration and forearm sampling and used our proximal tubule data that we derived from our MPS to populate. Oh, no wonder everybody's having problems with this. Use that data to populate this compartment of a 35-compartment PBPK model. This is the whole body at the top, and then this is just the renal side of our PBPK model that divides the kidney into multiple segments. So when we modeled our data, populated by an MPS model with actual clinical data, here's what we found. So the blue dots are patient-derived data from healthy patients who were administered IV morphine. And when you see our model in red and our patient data in blue, you see that it overlays quite nicely. And this is true for both morphine and morphine 6-glucuronide metabolite. So these were at doses of 30 to 60 micrograms per kilo, which is a somewhat subclinical dose. So of course, as I said, we already know what happens in drugs in healthy patients, but there's not a lot of data for pharmacists like me to guide our dosing decisions in patients with organ impairment. So what about our patient with kidney disease? We know a little, but we don't know nearly a lot. So thanks to Dr. Isaharanen and Dr. Huang, they also created a CKD PBPK model. And this is a complicated thing to do. Typically, when we dose drugs in patients, we scale the dose based on the reduction in GFR, which doesn't make a whole lot of sense because most of the drugs we're dealing with are not cleared by filtration, they're cleared by secretion. So this proportional scaling doesn't always work very well, particularly in drugs that are secreted actively by transporters like OAT1 and 3 or OCTs. So what they did was they developed an adaptive PBPK model that adapts the flows based on estimated blood flow. And this is data that was generated in animal models with some human data, and basically has been sort of an iterative process to try and define what the blood flows are within the individual segments of the nephron. They tested it tested and refined using something like 20 different compounds to derive this kidney disease model. So here's how we did using the CKD model. Again, this is the renal clearance of both morphine and morphine 6-glucuronide. Remember that in the kidney world, healthy GFR, so a healthy patient is on this side of the x-axis and the diseased patient is on the left side of the axis here. So as kidney disease progresses, that renal clearance declines, as we might expect, but it doesn't decline linearly, as you notice. So we also modeled morphine and morphine 6-glucuronide exposure in patients with end-stage kidney disease compared with patients' actual patient data. And again, the patient data is in the blue dots and our model is in the red solid or dotted line. We did okay. I think we could do a lot better. We particularly missed the mark a little bit here at the far end of the time concentration curve. But we're working on it. We think we can do better. So this is the roadmap. We filled in all of the gaps. We started with the MPS platform. We scaled that into a human renal clearance prediction, and then we used that human renal prediction to model the pharmacokinetics and the total exposure of drugs in actual patients in both health and disease. And we hope that we can use this model going forward to both help accelerate clinical trials and as well as a clinician to use medication better and more safely in our patients with kidney disease. So I want to acknowledge the team. It has taken truly a village to get this work done. It's the work of many, many graduate students and postdocs and generous funding by NIH and CATS and NIDA that has helped to fund some of the morphine work. With that, I'd like to take any questions.